Hello everybody, my name is Cristina Perez and I will be presenting my research on fMRI data analysis. First of all, what is fMRI data? Functional magnetic resonance imaging, or fMRI, has allowed us to peek into the human brain in action without the need for an invasive procedure. The most common method of fMRI uses the fact that when neurons in the brain become active, the amount of blood flowing through that area increases. So the activity-related surplus in blood flow caused by brain activity leads to a relative increase in local blood oxygenation. We call this the BOLD response. As the subject lays in the fMRI machine, they perform a series of tasks while 3D images of their brain are being constructed, usually about every 2 seconds, giving us images with relatively good spatial resolution and good temporal resolution. So the fMRI data is a 4D sequence of 3D volumes as images of the brain, where the fourth dimension is time. The BOLD signal changes in areas of the brain that are required to accomplish a task, and the goal is to identify the locations of such areas. There's, however, no straightforward way to identify the locations, as many problems arise. One of the problems is that the changes in task-related BOLD response are much smaller than the spontaneous fluctuations of BOLD in the brain, and it is necessary to present a stimulus multiple times to the subject to get sufficient statistical power to detect the task-related BOLD response. There is a need to remove the noise sources in order to retrieve the relatively small BOLD signal component. To remove some of the known noise, such as motion, pre-processing is performed. Pre-processing affects the data and how it will be analyzed. There is, however, no consensus on how it does and what is the best pipeline. It is also important to note that the response is sluggish and it is delayed by 5 to 6 seconds. The two approaches we have explored are the general linear model, or GLM, and the independent component analysis, or ICA. GLM is a very commonly used and it is available in toolboxes like FSL or SPM that are very popular amongst neuroscientists. GLM is based off a generalized linear model, and it is a massive univariate method where the same model is fit to each voxel in the brain, where a voxel is a 3D volume of about 3 × 3 × 3 millimeters. This method requires the parametrization of brain's response to stimuli and relies on assumptions of the distribution of errors and the shape of their variance. The further the underlying true model is from the assumptions, the less accurate the predicted activations map will be. ICA is becoming more and more popular. It is a data-driven method that decomposes complex multivariate spatial temporal data into independent subcomponents. This is a multivariate method that takes a V times T matrix, where V is the number of voxels and T is the number of time points, and looks at all the voxels simultaneously. ICA assumes a generative model where the observations are linear mixtures of independent components with estimated time courses that correspond to the time course of the stimuli are task-related components. By mapping this signal into the brain, we get an activation map that is associated with a task of interest. The data we use is called BOLD 5000 and comes from an open source dataset in OpenNeuro. The dataset is a large-scale, slow event-related fMRI dataset. The experiment showed about 5,000 images. Images were presented for 1 second with 9 seconds of fixation between trials. Participants were asked to judge whether they disliked, or were neutral about the image. There were 4 participants total, but we only investigated 1 of them to remain in native space. We investigated the contrast between 2 tasks, fixation versus image viewing. And here are some of our results. For the GLM, as heavily supported by literature, we have found activation in the occipital lobe associated with image viewing. And for ICA, I have 2 components here, where the first one is noise, and the second one shows activation in the occipital lobe and is associated with the task. What is next? To continue the comparisons of GLM and ICA, We want to compare the activation mass across 10 runs. We also want to simulate the data and see how accurate the predictions of each model are. We also might take a look into different contrast of tasks. That was all for today. Thank you very much for listening to my talk.